Great. And we're on. So this is the introduction to protocols.io. If you're already someone who's been using the site for a while, you might want to check out one of our regularly scheduled more advanced webinars. So you can take a look at protocols.io/webinars and all the different webinars that we offer on a regular basis. This is one for people who are really new to the platform and just recently created an account, haven't really played around with that. And I will go over the agenda in a second. As I said, we are recording this. We will send the link to all those who registered and are here, both to the recording and to the slides that you have, so you don't have to furiously try to screenshot or scribble notes. There will be a Q&A at the end, but as we go along, feel free to click on the Q&A button in the Zoom control and add your questions, and then we'll go over them at the end. It will be about half hour of me presenting and then 10-15 minutes for question answer. So it's not a full hour webinar. And if there are things we don't get to, as I said, please take a look at our other webinars, and also note that at this following link /help/demo, you can actually, if you have a specific question about the editor, about run functionality, whatever it is on protocols.io, you can request a one-on-one demo for anything that we don't cover today, or as you use the platform as questions come up, and connect with someone on our team for a 30-minute Zoom call where we'll answer questions, share the screen, and take you through demo as necessary. So that's the housekeeping. And on this webinar, as I said, this will be introductory. We're going to talk a little bit about the motivation behind creating protocols.io, why it exists. And then I'll switch out of slides and go over some of the key functionality in a live demo, protocols.io, and we'll touch on all of these things in terms of navigating the platform, and then come to the Q&A. All right. And so without further ado, let's jump in. And I like to start with this tweet from a scientist on the left-hand side, from a biologist at UC Riverside, who is complaining, I'm looking for a protocol in a 97 paper, as described in 96, finds 96 paper, is described in 87, finds 87 paper, paywalled. And this is at UC Riverside, so even UC doesn't have subscription to everything at the University of California. And of course, this is very common and frustrating experience. In the middle is a tweet, similar tweet from a physicist, which says devices were fabricated as previously described, and the original reference is devices were fabricated with conventional methods. So good luck trying to follow up on that and reproduce that method section. And I have a whole long collection where I've been bookmarking tweets and complaints like this. It would take us the entire hour just to go over the tweets that I have in the thread. For this, there's a link to them if you want to have a laugh in the slides that we'll share, as I said, later. But of course, this is a challenge when it comes to method sections of papers, and to make it a little bit more quantitative, we like to spend a few minutes discussing the Cancer Biology Reproducibility Project, which was a 2015 $1.5 million initiative of Center for Open Science and Science Exchange, which chose 50 cancer papers in top journals. And the idea was to independently replicate some of the key experiments and make sure that we can get the same results in a different laboratory. So they've set out to do that with 50 papers, and a year or two into it, when the first results were published in eLife, there was a lot of attention to it, a lot of media coverage, and Ed Yong wrote a nice long article about this in The Atlantic. And one of the parts in it was, the hardest part by far was figuring out exactly what the original labs actually did. Scientific papers come with method sections that theoretically ought to provide recipes for doing the same experiments, but often those recipes are incomplete, missing out important steps, details, or ingredients. In some cases, the recipes are not described at all. Researchers simply cite an earlier study that used a similar technique. And this obviously is similar to the tweets that I was showing. This is about published papers, but what is particularly interesting about this project is that the group doing this actually contacted the laboratories that did the work and collaborated with them, and they try to find the specific protocol. So it's not just about the published paper, but it's hard. Nope, I didn't want to open the article. I wanted to advance. It's hard to, even when you're collaborating with the authors that did the work, it's hard to find those protocols. Again, because the people move on, people forget where they wrote it down. They have 10 different versions of the protocol corresponding to result that they're not sure which one exactly it is. And so reporting on this, Center for Open Science, they have a page about this initiative, and they said that 0% of those 50 published papers contained the detailed protocols. And at the end of the day, they wound down the effort at just 18 out of the 50 papers that they set out to do. And a big part of it was the difficulty in actually finding the precise protocols that corresponded to the results. And the reason this is so striking is that we always think about the insufficient method details, like in the tweets that I showed above, in the published papers. But the reality is that the papers are sort of this external reproducibility, but the challenge with repeating work comes long before the publication inside our teams, inside the laboratories, in terms of how we're tracking, how we're organizing the methods, and that's what I call internal reproducibility. And so the mission of protocols.io is to sort of touch on all stages of the research cycle, not just on the publication. So our mission is to make it easy to share those method details as you're doing the work, before, then during, and after publication. And my own personal reason for getting involved and co-founding protocols.io is that as a postdoc, after my PhD in Berkeley, I was a postdoc at MIT, and I spent literally a year and a half of my postdoc tweaking, correcting a single step of the RNA microscopy, single cell microscopy protocol that I was using. So instead of 1 microliter of a chemical, I needed 5. Instead of a 15-minute incubation, I needed 1 hour. And at the end of the day, it's not a new technique. It's a correction of something previously published. And that means that I don't get any credit for that year and a half. And everyone else who is using the same technique is either getting completely misleading single cell results or has to spend a year or two rediscovering that correction. And so I became sort of obsessed with creating a central place where even after you've published, you are able to share corrections and optimizations. So at a glance, we launched protocols.io back in 2014. And speaking of that internal and external reproducibility, there are 2 sides to protocols.io. There is the public-facing side for people to openly share sort of Wikipedia style crowdsourcing the method knowledge. And that part is open access, CC BY. It's free to read and free to publish. You don't need a subscription for that. And it's for the internal reproducibility when people use protocols.io in their company, inside their academic lab, that's when we charge for that internal use. And so at this point, we have just over 130,000 registered researchers on the platform, and there are almost 15,000 public protocols that have been shared, and over 60,000 private protocols that people are either keeping and organizing for their own work, or more frequently sharing with their colleagues and collaborators, co-authors. And to the public methods, each year there's over 1 million people that come to view the public protocols that scientists are sharing. And so that's where we are now, and I'm going to now skip out of the slides and transfer to the platform itself. Right, and I'm going for the live demo for the introduction. I'm going to show one of those 14,000 public protocols that we have, but it's important to note that when you're on protocols.io, every protocol you create by default is secure and private. It's yours. You decide who to share it with, whether to share it at all, if you share it with anyone, what permissions, access permissions to give to them in terms of viewing only, or viewing and editing, or full collaborator who can publish and is a co-author. But you decide who and when to share it with. You decide if you want to make it public. That's a very explicit action, and there's a publishing flow, but by default, everything is private and yours, and you're in control of how and when you share it. And in terms of the functionality, what I want to highlight is that these are not static, fixed PDFs. It's not a published paper done and fixed. These are dynamic and interactive. So what I mean by it, it's not that it changes constantly and you never know if a protocol you looked at yesterday is going to be the same today. But what I mean by these being dynamic and interactive is you can include videos inside the steps, tables with formulas. There's a lot of rich functionality on protocols.io around using your own or other people's protocols. We'll talk about in a second. You can also notice that as you mouse over the different steps of the protocol, there's a little plus sign that appears on the right-hand side that lets you actually ask a question that will go to the authors and to everyone who's using this protocol. And so as an example, on step 9, there are 2 comments. If we go to step 2, you can see there are 7 comments that are happening right on the step. Again, just like protocols themselves, you decide whether you're asking something publicly or privately, even if it's on a public protocol. But when the comments and discussions are happening in public mode, you're sort of building up an FAQ right on the steps of the protocol that everyone can come to and see without having to email the authors and without you as an author having to answer the same question over and over. You can point people to the answer, or hopefully over time, they see the answer in protocols.io and don't even email you for it. And as these discussions happen, we frequently see that scientists will create a new version of the protocol. So with the click of a button, you can say, make a new version, and then you're able to edit it and publish that. It'll get its own DOI. And there is a compare button that actually lets you, with a click, see a glance of what is the same and what's different between the versions. We'll highlight the steps and what has changed, and you can see what's the same, what's different between version 1 and version 2. And so this is the part that lets you keep your protocols up to date. Once you publish it, it is fixed and everyone can get to it in perpetuity. but you can come back and you can share those corrections and optimizations. Moreover, there is this copy fork button, and that actually lets you treat every single protocol, whether it's public or private, as essentially a template. So any protocol that you see on protocols.io, you can click copy fork, it'll create a clone of it that becomes private to use. So if you're working with kidneys and I'm working with lung tissue, I can take a kidney protocol and maybe I have slight variations, slight tweaks on it. So I can copy a protocol and then I get the edit button, it becomes private to me. I can make changes to it. So all of these protocols are reusable, extendable, people can build on them depending on the equipment in our research teams, depending on the organisms that we are studying. And then the same compare functionality that I just showed between versions, that applies between forks as well. And if you want to share your tweaks or optimizations, you can easily do that. And we tie the protocols to each other. You can see there's a forks tab. So from the original or from the fork, you can go back and forth, and we try to give credit and make it very clear what's the original, what's the fork. And we try to give credit both to the people that have originally shared it and to those who are sharing different versions of it or optimizations. All right, so the idea is to create a place where you can easily build and reuse, build on and reuse the protocols that are in the repository. You'll also notice there's a works for me button. So on protocols that have a lot of engagement, if it works for you, you can click it to give it sort of an upvote. We don't let you vote down, but over time, protocols build up a sort of signal of trust from the community. We don't peer review the protocols, so it's a crowdsourced platform. These are your protocols that are being shared, but we do try to share, when you look at the metrics tab for the public protocols, we try to share everything we know about the engagement with and use and reuse of these protocols. And the last thing I wanted to say on the protocol level is that there is a run button that's particularly useful for hands-on techniques. So of the 14,700 methods that I mentioned, I think somewhere around 90 to 95% of them are wet lab methods, 5 to 10% are computational, and you're very welcome. I'm a bioinformatics scientist and experimental scientist. So you're very welcome to share computational pipelines and workflows on protocols.io with links or attachments inside the step of your code or data and links to GitHub. But for the protocols, for the majority of protocols and protocols.io in the public repository that are hands-on, there is a run button that allows you, as you're performing the experiment, to use protocols.io sort of as a cooking checklist app. So you can see where you are, what you're doing next. If there is a timer, you can start it right on your iPad or Samsung tablet in the Fume hood. And importantly, as you're going through the experiment, you can click edit and you can say, today I used 4 microliters instead of 5, 30 degrees instead of room temperature today. And then when you're done, you'll save it in your folder, organized, that I'll show in a second. And you have a record of how exactly you use that protocol on a particular day. You can export it, you can put it into an ELN, print it out, paste it into your physical lab notebook, or simply keep it on protocols.io for reference. So you're not changing the underlying protocol, but you're creating a runtime record of how you perform the experiment on that particular day. So it's something that should hopefully save you a little bit of time as you are doing the experiment. So again, the run button is for the hands-on and wet lab and field methods, right? And sort of finishing up in the live demo part, I also wanted to say that When you create an account in protocols.io, once you sign in, you will spend most of your time in the file manager. So this is where I am right now. You can always switch to it in your profile. So out of the public repository, I just switch to the file manager. And this is where I have workspaces. And this is where you create folders, subfolders, organize your protocols. And if you're using protocols.io for internal development and organization with your collaborators or teams, this is where you create a workspace. An administrator can invite members and everyone is sort of on the same page, using the same space to share the protocols across the company, across the organization, or within a team. And their typical workspace might be 5 to 10 people. We also have workspaces with hundreds of people in them. So there are consortiums, there are collaborative groups that are working all across the globe. And they use protocols.io to communicate and collaborate with each other. Not as a lab notebook where they're sharing all of the pages, but specifically for organizing and collaboratively doing method development. And if you're new to protocols.io, I want to point out that very frequently, we come in with already most of the time, unless you're an undergraduate or rotation student who is just joining a lab, you're typically someone who already has existing protocols. And so I want to spend 3-4 minutes just talking about the different ways to get existing protocols into protocols.io. When you click, so here's the new button. When you click create new protocol, you'll notice that you actually have the ability to upload files. So kind of like Dropbox, you can put a PDF, a zip file with existing protocols, Microsoft Word, PowerPoint. You can just put it into those folders, into the workspace. So you can see here, transformation.pdf. I did not create a new protocol from scratch, but I just dragged and dropped the file into the workspace. So that takes you 10 seconds. That takes you 10 seconds, and it's not taking full advantage. It's not taking advantage of the rich functionality of the editor, collaborative, concurrent editing that we have, all of the components that add structure and the details that are necessary for you or someone else to repeat the experiment. It doesn't take advantage of that, but it is a quick way to just take 30 protocols that your research team uses and just quickly get them into central space. However, if you have 2-3 minutes, what we recommend, again, if you're short on time and you want to quickly get existing things in, our recommendation is to say, make a new protocol. You choose where it's going to go. So demo group, continue. And instead of just dragging and dropping the PDF into the file manager, what I recommend is creating a protocol. Here's the look inside the editor, but we're not going to add steps and take 30 minutes to put the full existing protocols in. All we're going to do is give it a title, spend 1 minute adding abstract, I can't spell anything when I'm doing a live demo. Add an abstract, maybe some keywords, and then scroll down on the description tab, scroll down, click add attachment, and drag and drop your PDF right into here instead of just dragging it into the workspace. And so what is the point of doing that? This is a little bit better than just dragging and dropping in the PDF. If I go back to the file manager, here is that test protocol that I just created. Let's reload. So there's our test protocol. And the difference between this and the PDF is if you have many protocols and folders and subfolders, this is searchable with the keywords, the title, abstract that you put in. So when you come back in 3 months, or if you have multiple workspaces that you're part of, taking that extra minute or two per protocol, per existing protocol, to add the attachment to it, makes it much easier to retrieve and find, so they become much more discoverable within the workspace. And the other thing that you'll notice on it when you click on it inside the file manager is that not only can you view and edit, but you now have a publish button. So even though I didn't put in a full beautiful protocol taking advantage of protocols.io, I can actually quickly make this public as an attachment. It will get a DOI. It's not perfect, but it is a way to, if you need for publication or for sharing, or you want to get all of your team's protocols quickly out without taking 30 minutes or an hour for each one, this is a way to very quickly get that knowledge out into the world. So that will give you a DOI. That is something that is then discoverable, and people can find it through Google and navigate to it. And remember, because we have versioning, you can always come back. You can always come back, create a new version of the protocol, and instead of just an attachment, add the beautiful step-by-step and component formatting to the protocol. And coming back to this, I also wanted to say that if you are someone who does have a few extra minutes, let's say 10, if it's a simple buffer, if it's a simple protocol that you don't want to just add as an attachment, the third option obviously is to enter protocol using our editor. But you can, of course, create individual steps, copy, paste. But what I also want you to know is notice that inside the editor on the steps, there is a button which says if you have an existing protocol, you can insert steps from a text file. So if you have a Word, Google Doc, you can click that, paste the steps into here, and it will convert it into the step-by-step format. And then, again, you can still add the attachment, you can publish it, you can start gradually adding components, formatting, and make it a product that is easy for you and anyone else to really learn from. You can take a phone, go into the cold room, make a 10-second video of how you're preparing the sample, add it into that step. So, of course, that does all require more time, but the easiest is drag and drop, then create a protocol shell, title, abstract, keywords, and add the attachment. Or you can pull the step-by-step information into the protocol easily with this insert steps from a text file. Right? And don't try to make it perfect. Again, there's versioning. You can always come back. You can always take feedback and questions and create version 2, 3. There's some protocols that are in version 15 if people are really actively doing method development. So you can always clarify. You can always share the improved version of the protocol, even after you've made it public. And I also did say that we don't peer review. We screen all protocols to make sure that if someone is publishing, we screen to make sure that no spam, no pseudoscience goes into the repository. But we don't peer review a protocol. We don't go into the lab to test it, but you'll notice that there is a button on your protocol. If you're an academic scientist, there is a button that says peer review options. And this is a partnership that we have with the journal PLOS ONE that lets you easily send your protocol for peer review in PLOS ONE. And they created a separate channel called, a separate article type called Lab Protocol within PLOS ONE that is not for research articles with new results, but specifically to ensure that people get credit for method development and the work that they're putting into tweaking and getting something to work in a new species or cell line. So take a look at those things. We have a whole separate webinar called Getting Credit and Recognition for Your Method Development, where we talk about the public sharing. But the last thing I wanted to say for the private protocols is that there is, on every single one of them, there's a share button. So short of making it public, you can always click the share button. You can invite people by email, individuals, and you can decide what kind of access they will have. So this will send an email invite, and then you can select what permissions this person has. You can also attach your protocol to a workspace or multiple workspaces to give all the members of a specific workspace access to it. And finally, at the very top of that share link, you'll see that there is what we call a reviewer link. So this is a private link to the protocol that editors, reviewers, or anyone else that you want to get access early to the protocol before you make it public if you're academic. This is a link that you can share, and it does not require other people to have registration or an account on protocols.io, so they can easily get to it. And if you're submitting your work to a journal that has double-blind peer review, we also have a link that will strip all the author and affiliation information out of the protocol, so you can include it in the publication. Right? I am going to now exit out of the live demo mode. We've gone through many of these things. In terms of existing protocols, one thing that I did not mention is that we also have editorial service where you can come in, and once you sign into protocols.io, you can actually go to We Enter Protocols and drop your PDF or Word document in here. And we do charge for it if you're not on a site license campus-wide plan. We do charge for that, but this is something that lets you send your protocol to us. One of our editors with science background will put it in. Another editor checks it to make sure that there are no errors, and then we privately transfer it to you. We notify you when it's complete. If it looks good, we transfer it to you. It's not an expensive service. Starts at $50 per protocol, but if you're in a premium plan already, then it is included for many of you. And so if you go to protocols.io/institutions, if you scroll down there, you will actually see the list of organizations and universities. If you're in a private pharmaceutical or biotech, we don't have you in the list, but if you're at one of the universities or at FDA, or you're part of a funder like ASAP Parkinson Funder or More Foundation, then you have premium protocols.io, and that protocol import is actually included. So if there are 10 people in a lab that are on protocols.io, you can send us 10 existing protocols for free, and we'll put it in if you're at one of these universities or are funded by one of these funders. So there is a list there. And going back into the slides, I think I've gone over most of the functionality. These are my backup slides in case the live demo is not cooperating. And I wanted to finish up by saying that there are many journals. So I think it's actually over 600 journals right now that explicitly have protocols.io in their author guidelines. When you submit a paper, they encourage you to add a link from the method section to protocols.io. And more funders that recommend or require their grantees to share the protocols and explicitly encourage use of protocols.io. And as I just mentioned a second ago, there are more and more universities that since 2019 have been both universities, government agencies and industry organizations that have been signing up for site licenses that make protocols.io premium and unlimited for everyone at the organization. And this is, as I said, where you can check if your institute or organization has a site license. And with that, I am going to stop, as I promised, to make sure that we have time for questions. And again, thank you for taking the time out of the day to learn about protocols.io. As a reminder, you can always send us your questions. You can hop on a one-on-one demo for specific questions as you use the platform. And our response rate is really good. We have excellent customer support. So if you email info@protocols.io, usually in a couple of hours, you'll get an answer to your question. So if you don't get to ask something today as you start using protocols.io, you will always have the opportunity to get in touch and jump on a call or ask us something by email. All right, and with that, I will stop recording